Hello, the purpose of this screencast is to provide a mini lecture over Chapter 4, Classical Conditioning, Learning to Predict Important Events. So classical conditioning, we can think of some thought questions to kind of preview what we're about to learn. First of all, I'd like you to think about what is classical conditioning. Then think about what are the rules for classical conditioning, meaning under what conditions can classical conditioning occur. And then think about, well, how can we explain this phenomenon of classical conditioning? And the purpose of this mini lecture is to help you answer those questions. So the learning objectives of this chapter, I want you to be able to identify and define the basic terminology, know what an unconditioned stimulus is, an unconditioned response, be able to identify the conditioned stimulus and conditioned response in a classical conditioning situation. We will review how Pavlov demonstrated classical conditioning in dogs, and so I'll expect you to know that. We'll also talk about appetitive and aversive conditioning and approach and avoidance responses. So I'd like you to be able to distinguish between those. I want you to understand the difference and be able to describe the processes of acquisition, that is, initially acquiring or learning a conditioned response, as well as extinction. We will also review conditioned emotional responses, or CERs, as well as conditioned compensatory responses, CCRs, and we will talk about how those relate to drug tolerance and overdose. And then I want to review with you several blows to conditioning theory, and we'll talk about their theoretical significance. Okay, so let's begin. Some key ideas as a big picture overview. Classical conditioning is the same thing as Pavlovian conditioning. We use these terms interchangeably. We can abbreviate CC, capital, for classical conditioning, or PC for Pavlovian conditioning. I'm kind of conditioned myself to prefer PC. So if you see PC anywhere, I'm talking about Pavlovian or classical conditioning. Some other big picture ideas. All organisms, even tiny little sea slugs we will talk about, can be classically conditioned. Classical conditioning happens automatically and without conscious awareness. That's really important when we think about the significance of all of these conditioned behaviors that we have. Classical conditioning is one type of associative learning. We just got out of our chapter on habituation and dishabituation, talking about them as examples of non-associative learning. Because in those kinds of non-associative learning, there's only one stimulus. And in associative learning, there are two. So we are learning in classical conditioning to associate two stimuli. And because of that, it is sometimes called SS learning or stimulus-stimulus learning. And what are the stimuli we're learning? Well, we are going to learn that they're called the conditioned stimulus and the unconditioned stimulus. So how does it work, classical conditioning? Well, unconditioned stimuli are naturally occurring stimuli that elicit innate or inborn reflexes. For example, food is an unconditioned stimulus that automatically elicits salivation. When food is put in your mouth, you automatically salivate whether you want to or not, and you don't have to learn that. It's a reflex. Another example, puff of air to the eye will cause you to blink. If I blow in your eye, you're going to blink. If you experience what my daughter loves to call the jump scares, right? Something scary or painful, we automatically jump, or animals will freeze. It's a protective mechanism so that if it is a potential predator, maybe the predator won't see them. So another example used in the book was the sexually available female quail. And so, of course, that would be the unconditioned stimulus to elicit mating behavior in a male. You can think of emotions getting like fun times being unconditioned stimuli for like a happy emotion and so forth. So seeing the words is a little bit more difficult. The take-home message that I want you to be able to see is, I think, illustrated better in the graphic. Unconditioned stimuli cause, with this arrow, unconditioned responses. Food causes salivation, air puff causes blink, something scary or painful causes fear, females cause a sex response, fun or good times cause happiness, and none of this is learned, right? That's why they're unconditioned. These are all innate reflexes that we have. So the unconditioned responses, again, just to be clear, are the reflexes that get elicited. If we look in our graphic, all of these responses or reflexes are the URs. So unconditioned stimuli, naturally occurring stimuli that elicit or cause these unconditioned or natural responses. Again, nothing yet is learned. Unconditioned stimuli cause unconditioned responses. So where does the learning happen? Well, the learning happens with these conditioned stimuli. So conditioned stimuli, or CSs, are any stimuli that might get associated with or paired with the US. So in Pavlov's experiment, he associated a bell with food, and eventually the dog learned to salivate to the bell. Some other examples that are talked about in the book are using tones or lights paired with air puffs to the eye, or shocks, or quails, female quails in heat or whatever. You might be able to relate as a little kid hearing the jingle of the ice cream truck, right? That becomes a conditioned stimulus that ice cream is available, or fun or good times are available. We may learn that 6 o'clock means dinner. You may learn, taking courses on campus, seeing your professor with a stack of papers signals that a test is coming. So we have all these examples again. I'd love to map them out graphically. I feel like that's a little bit clearer. In Pavlov's experiment, food was the natural unconditioned stimulus that caused the unconditioned response of salivation. After several pairings, this bell, just a neutral stimulus bell, if it reliably predicted that the food was available, it was learned that the CS now became a conditioned stimulus, and then the learned response became salivation. I know we didn't get to the CRs yet, but let's look at a couple more examples of that, right? We've been talking about how food, air puff, good times. I have food here again. 6 o'clock. Oh, for the 6 o'clock dinner. To put it together with the signals that we saw in Pavlov's experiment, a bell was associated with food. The bell came to elicit salivation. Tone or light was associated with air puff or shock, and in those cases became to elicit blank or pain or fear. The truck jingle becomes associated with good times to elicit happiness. 6 o'clock is associated with food or dinner, elicits hunger, salivation. Stack of papers becomes associated with a test, starts to cause anxiety, and so forth. And so then the conditioned responses are those learned reactions. Where is the learning coming from? It's not like learning a brand new reflex, right? Notice, and we'll see this on the next slide, these responses or reflexes are the same. It's learning to do the reflex in a new situation in response to this new learned or conditioned stimulus. So think about that for a second. There's a graphic in the book. I find it extremely confusing, so I just wanted to explain it. They're showing that without learning, this dog, the unconditioned stimulus of food will come to elicit this UR of salivation. Here we have a CS bell is introduced, and in the beginning, the dog is still just salivating in response to the food because they're happening together. Eventually, take away the food and the dog will drool to the CS bell. But the take-home message here that I'd like to spend a minute talking about is the fact that the UR over here and over here is the same as the CR. And in my drawing or illustration, the UR and the CR are the same. The difference is the eliciting stimulus, right? The UR is salivation to the food, whoops, and the CR is salivation to the tone or the bell. So that's a quick overview of classical conditioning in general. I do want to keep my lecture here pretty brief. So let's talk about a couple other critical things. The take-home message I want you to have here is that classical conditioning is adaptive. What do I mean by adaptive? It helps organisms to prepare for important biological events. For example, if dogs could learn to salivate before food enters the mouth, it could help prevent choking, it aids with digestion, and the same for humans. If I know to get ready to eat at 6 o'clock, I can start salivating and start preparing my digestive system for that food. If I could learn what predicts dust about to blow in my eye, I could blink before it gets there. If I could learn what signals mating opportunities or good times or fun or eating opportunities, that's going to be good for me to be able to approach those before they go away. And also, if I can learn what predicts danger, shock, pain, these jump scare events, I could avoid them before they happen, avoiding danger. So classical conditioning is adaptive. The other thing your book makes a big point about is that classical conditioning is ubiquitous. That means it's found everywhere. And they give some examples about how all multicellular organisms can learn it. We know humans, rabbits, rats, mice, all mammals, and we use them all the time in studies of classical conditioning. The book talks about quail and other birds, flies and other insects, and then, of course, the aplysia, which is mentioned with respect to that gill withdrawal reflex in fear and aversive conditioning. So, classical conditioning, adaptive, protects organisms, and therefore is ubiquitous. All organisms can learn it because it helps them to adapt to their environment or protect or prepare for events. So the next section in the book is talking about appetitive versus aversive conditioning. So classical conditioning is divided into these two main types. Appetitive, where the US delivers something good or desirable or pleasurable. So if you are conditioning to food or sex or drugs or shopping or gambling, those are considered appetitive types of conditioning, contrasted with aversive conditioning, where they're using things like shock or puffs of air to the eye or exposing you to allergens or poison or bacteria or a virus, making you throw up. Those are aversive situations. We learn both. Again, why? Because the appetitive conditioning opportunities lead to approach, right? And the aversive learning opportunities lead to avoidance behaviors. So again, they help us to approach good opportunities, mating opportunities, eating opportunities, and so forth, and avoid dangerous opportunities. Okay. Here's a graphic from the book. It's just outlining a few of the varieties of classical conditioning. I do like because it does break it down into appetitive versus aversive. So you can take a look at that, and then it does detail the US's, URCS's, and CR's. So I do like that graphic and draw your attention to it. The book does mention these CERs, or conditioned emotional responses. We do know that emotional responses can be classically conditioned, like happiness. Like when you hear a song and it reminds you of your prom and you feel like, oh, that was great. Or maybe you broke up with your partner at prom and you hear that song and it makes you angry, right? So we can definitely condition our emotions. And John Watson did this classic study to show how, like, most phobias are probably classically conditioned responses. We're born with very little fear. And so in this classic example, and of course there's a video on Canvas you can check out that goes through the classic Little Albert experiments, showing how little Albert before conditioning had very little fear. He did not fear rats or rabbits or old men with masks or monkeys or dogs or anything. But after classical conditioning, where little Albert was playing with a white rat and they repeatedly presented this loud, frightening noise that startled him, The loud noise naturally produced fear. Albert began to associate the rat with the loud noise and then became afraid of all rats. We also talk about little Albert in the context of generalization, where even though he only had the bad experience with a rat, he became afraid of like anything white or fluffy. And so we call that generalization. But that's pretty straightforward, I think, in the book. Some concepts that I think do get a little bit dicey and hard to understand in the book is this next one, the conditioned compensatory response. So for the most part, like we saw in a previous slide, the UR and the CR are identical, right? And most of the time, 90% of the time, that's true. Sometimes, though, when we have a particularly important biological event, the CR and the UR are the exact opposite of each other. For example, they're using the example in the book of how if you inject adrenaline, it automatically increases heart rate, right? Adrenaline, epinephrine automatically increases heart rate. If you continually inject a rat with the same adrenaline in the same context, like they talk about that testing chamber, then pretty soon as you put the rat into the test chamber, it's like getting ready for that injection, and its heart rate will decrease, like significantly, in anticipation of the increase that's coming, right? That adrenaline, it's like your body's saying, uh-oh, my heart rate's going to go up. I don't like my heart rate to elevate like that. I'm going to bring it down now so that when I get that injection, my heart rate will be normal. It's based on this concept of homeostasis or biological balance, right? So having that opposite reaction, decreasing heart rate in anticipation of that impending increase, helps to keep the body in balance. So when you get that adrenaline, your heart rate isn't so elevated. It's another example of how adaptive classical conditioning is, right? We're learning to compensate for the negative effects of potential stimuli like these repeated injections of adrenaline. What I'd like to do now is take this a step further and talk about how this relates to drug tolerance and accidental overdose. So let's take a drug, for example, heroin. Heroin, unlike adrenaline, actually decreases heart rate. That's a main effect of heroin. Well, when people start repeatedly injecting heroin addicts tend to shoot up in the same way. They tend to shoot up in the same place with the same people because these become safe. It's obviously not a good idea to, you don't see a lot of people shooting up heroin and whatever. So people will become conditioned, use these stimuli, the needle, the room, the location, the people, the way that they're mixing their drug and injecting, will all become conditioned stimuli that the heroin is coming, and the body will respond automatically, unlearned, without our conscious awareness, to increase heart rate. And I bet a lot of heroin addicts perceive that, right? And they just think of it as like, oh, I'm excited to get the drug. It's my heart is racing right now because of anticipation. And really, it's a conditioned compensatory response. It's the body getting ready to like, uh-oh, my heart rate's going to decrease. I don't like that. It takes me out of balance. Let's increase heart rate so when the heroin comes in, now I'm back at a normal level. So that is actually what's motivating tolerance, right? That the body is physically preparing itself for the drug. And so now we need more of the drug to have the same effect because the body is actually rebelling against the drug. That can also explain overdose. In fact, there are many this goes back to the 1970s when Shepard Siegel was first studying this phenomena of people who were seasoned addicts who did not have tainted supplies of drugs, were overdosing, and immediately, like with the needle still in their arm, suggesting that something crazy was happening here. And what he realized after many, many laboratory tests was that the unique, well, laboratory tests, but also evidence from humans, was that for these cases of human victims who were dying of overdose with untainted drug supplies and normal doses for them, there was a common thread. They were shooting up in a new place or with new people or in a new way. It wasn't that the drug itself was tainted or a new supply. It was that the context. So they were not getting these CSs of the room, the location, the people. Their body was not performing the conditioned compensatory response because it didn't know to expect the heroin. So as soon as they injected the heroin, heart rate decreased. And now, because they had been tolerant, their level was so high, the dose of the drug, technically normal for them, but was really a lethal dose for somebody who was not producing these conditioned responses. And so we learned that these CCRs are classically conditioned compensatory responses do play a very important role in drug tolerance and drug overdose. Okay, so I am going way over time here. I wanted to keep this very brief. I'm going to try to finish this up in like 5 minutes. The book does talk about like compound conditioning and overshadowing, and I don't want to focus too much about that, but what I do want to focus on is this whole idea of, is classical conditioning then just simple SS learning? Like, could we just pair any stimuli in the world with an unconditioned stimulus and learn that signal? And it turns out that the answer to that is no. And so I want to talk about these 4 blows to this idea that classical conditioning is just simple SS learning: blocking, latent inhibition, timing, and associated bias. So we always talk about Kahneman's blocking effect, or Kahneman's blocking paradigm. Kahneman was the researcher who discovered it. And what Kahneman discovered was that if, and this is a little bit tricky, so I'll take a second to try to walk you through it. If you have an organism who has already learned to associate a CS1 with a US, like a tone with a shock, they will respond to the tone with fear. If we then try to add a new CS2, like a light, with the tone, you would think, oh, no problem. The animal will easily learn to associate the tone and the shock, or the tone and the light, for example, and will start responding with fear to the light. But what Kahneman showed was that's not what happened. So I want to walk you through this blocking paradigm because it's very confusing. What it's showing is that there is a control group here. The control group is just showing that normally if we pair a light and a tone together first and then associate them both at the same time with shock, organisms will learn to fear both. And that's what this control group line is showing us. The rat is first conditioned at the same time with the light and the tone and the US shock, and they learn to fear both. But in the experimental group, they're trained first that the light predicts the shock. Then they add the tone and the light, and guess what? The animal does not learn that the tone also predicts the shock. Why? Why does that happen? To map it out for you, that what they're showing here is that in the first part of the experiment, they're paired with CS1, tone, and shock, and they learn right away that the tone means shock is coming, and they learn to be afraid of the tone. They then add the CS2 light with the tone and the shock, but the animal still does not learn that the light signals shock. Well, that's crazy, right? The light is here and the shock is here. Why don't they learn that? And that was a blow to SS theory, right? Because you have, here's a stimulus, here's another stimulus, here's even a third stimulus. They should all be getting associated. But blocking, they did not get associated. Why? Well, because, and we're going to talk about Rescorla-Wagner model in a minute, Rescorla-Wagner is suggesting that there's no surprise. There's no new information here. The rat already knows to expect the shock from the tone. They don't care about the light. They're so afraid of the tone already, right? They don't care about the light. And it's not even just fear conditioning. It happens with food conditioning too. They're so interested in the tone that's predicting the food, they don't care about the light. So adding that second stimulus is blocked from learning because it's not adding any new information to the animal. A second blow, and I'll try to run through these. These are a little bit maybe easier, but latent inhibition, I always learn this as the CS pre-exposure effect, which I think may help to clarify. Basically, what's happening in latent inhibition is that if the CS is already familiar, it will not be associated. So here, this is another confusing graphic from our book, but the control group just sits in the chamber, never gets exposed, and is then presented a tone and a shock together, and they will learn. That's why it's confusing. I don't know why they're called the control group. I guess just to show what happens without latent inhibition, but learning will occur, and they will learn to fear the tone if it's in phase 2. In the experimental group, what they're doing is they repeatedly present the tone to the animals, whereas the control group just sat without ever hearing the tone. So in the experimental group, they repeatedly hear the tone over and over and over. Then they try to associate the tone with the shock, and lo and behold, no learning. To map it out, in latent inhibition first, there is some CS pre-exposure time, right? That's why I always learned it as CS pre-exposure effect. So they're pre-exposed to the tone. Tone, beep, 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 nothing happens. Then they try to teach the animal that the tone is going to predict shock. The shock will cause pain and fear, but they will not respond to the tone. That's latent inhibition. Why? Again, Rescorla-Wagner model is going to be telling us, like, there's nothing surprising there, right? So the CS was already predicting that nothing was happening. It's very difficult for us now to retrain ourselves at the CS, or for the rat to retrain themselves, that the CS now means something different. It already had a meaning. It meant nothing. Safety, maybe. So trying to get it to learn to associate it with something else. I think a good maybe analogy here to real life is like, if your dog is used to seeing you put your shoes on and take it for a walk, it's going to be very difficult to get your dog to associate you putting your shoes on with just leaving the house to go to work. You know, they're used to those shoes meaning, hey, I get to go for a walk. Or maybe that's not a great example, but think about that for a minute. Okay. A third blow to conditioning theory is timing, meaning the order of presentation of the CS and US, and also the length of time between. The best way to explain this is through this picture. And here I want to look at this picture on the left. Ignore this graph. But basically, and then I want to show you some of these concepts. So in backwards conditioning would be where the US was presented first, like the food, and then the tone. And you know what? It doesn't work like that. So that even in my graphics where I'm drawing, let's go back to one of these, like even just with food. I have a food one back here. Hold on. Okay, so even in something like this, even though we're drawing the food first and the tone second, The tone has to actually happen first, because if the food is presented first, they call it backward conditioning. If the food is presented, the dog is already salivating and eating. They're not going to pay attention to the tone. So the tone has to function as a signal, right? The CS has to be the signal that happens first. It's like going through the intersection and then getting the red light. It's too late. You're already through the intersection. You're already in the wreck. You don't care what color the light was. So it's very important that the CS come first. If the US comes first, they call it backward conditioning. And it doesn't work. The animal will not learn. The best kind of conditioning then is called delay conditioning, which really confuses me. There is no delay at all between the CS and the US. I don't know why they call it delay conditioning, but that's what's being illustrated up here. In a typical conditioning situation, Pavlov's dogs, the light and the shocks, the quail, all of that, the CS is present, the signal. What my little thing isn't showing here. So the CS signal is on for quite a while, not like hours, but usually about 500 milliseconds, a half a second. Light comes on, hey, pay attention to the light, and then the food shows up. So light comes on first, and then the food shows up. And so they're kind of happening, the CS is happening first, and then the US shows up, and then they get associated. I will mention this, that if the CS and the US happen at the exact same time, the CS will not be learned. They call it simultaneous conditioning. And again, if they're there at the same time, the food is already present. Nobody cares about the tone. So the tone does have to come on first, and then immediately right after, or even at the same time as the tone, then the food shows up, then it will get associated. It's called delay conditioning, produces the best learning. In trace conditioning, this one, the signal, the light comes on, and then like some time passes, and then the food shows up. And it's just like with any kind of conditioning or association, they got to happen close in time. This can be learned, but it's pretty difficult because it requires that the organism remember. So why is timing important? Well, in backward conditioning, if the US comes first, the CS becomes meaningless. It doesn't provide a signal at all. In trace conditioning, that bottom one we were looking at, you have to hold that memory trace of the signal, which is challenging if nothing's happening. Who cares about the CS if it isn't signaling anything, right? Oh, I forgot about that old yellow light, or I wasn't looking at that anymore. It didn't mean anything to me. The last blow I want to talk about is this associative bias. And the key here is that some associations are just naturally easier to make. For example, Garcia and Kohling's study, what they showed was that rats could learn to associate a flavor with illness, but they could not associate a flavor with painful shock. Similarly, they could associate a tone with painful shock really easily, but they could not associate a flavor with painful shock. So, if tone and taste were both paired with poison, they only figured out that the taste signaled poison and vice versa. So why did that happen? Well, in nature, tastes go with getting sick, right? It's really important to be able to learn that, those taste aversions or conditioned taste aversions, which are another form of classical conditioning. You associate a flavor like maybe SpaghettiOs with getting sick or throwing up, and now you'll never eat SpaghettiOs again, if you've ever had that happen. And so this associative bias is showing us that, like, and think about that too with the flavor. Like, you might have been watching Andy Griffith while you were at home throwing up, or Friends or whatever, but you don't feel nauseous now every time you see Andy Griffith or Friends. That would be maladaptive. So we're not very good at associating environmental stimuli that aren't flavors with getting sick, because if we did, then we'd avoid the world around us. But it is super important for us to avoid flavors, right? Because evolutionarily, what if we ate a poison berry or poison milk or poison meat? We have to be able to learn to avoid those things. Okay, so all four of these were blows to SS theory. And I mentioned, I'm almost done here. I promise, almost at the end of my little mini lecture here. I have mentioned a few times Rescorla-Wagner model. It is so confusing to understand from the book, and I get that. What I want you to learn about Rescorla-Wagner model is, it is a mathematical model of error prediction. Why do we care about that in classical conditioning? I don't want you to know the math. I don't care that you know all of their contingency versus contiguity arguments. But what Rescorla-Wagner model is elegantly showing for us is that if the CS is not a reliable predictor of the US, then we're not going to learn the association. What that means is that if I only sometimes put my shoes on before I take my dog for a walk, the dog is not going to learn that the shoes mean go for a walk, right? So there has to be good predictability there. If probability or predictability, every time I put my shoes on, I take the dog for a walk and only take my dog for a walk. I never put my shoes on or those particular shoes on when I do something, when I don't take the dog for a walk, then that's 100% predictability, and the dog will learn every time I put my shoes on to get ready to go for a walk. And so based on, how predictable our stimuli are, variations of learning will happen in between. So I hope that does clarify Rescorla-Wagner model a little bit for you, but of course always ask questions if you're confused. It is confusing. Summary, take-home message, conclusions. What do I want you to know about classical conditioning in 38 minutes or less? Classical conditioning is an automatic, unconscious form of learning that affects all organisms. Why? Because it's adaptive. It helps us to promote survival. It helps us to protect ourselves. It helps us to get ready for important events. Classical conditioning allows for learning that novel signals, right? These things we're calling CSs can predict important events called USs. And while it is true, SS theory is true to some extent that just about any stimulus can become a CS, there are some limitations. The limitations are this: if the CS is not novel or surprising, it won't be learned. And we call that latent inhibition or the CS pre-exposure effect. If another CS is already present and doing a good job of predicting the US, a new CS won't be learned, and we call that blocking. If the CS is presented after the US or remote in time from it, it won't be associated. So like if I put my shoes on at 8 o'clock in the morning, but I don't take my dog for a walk until 4 p.m. that afternoon, they're not going to get that association. So timing matters. And then if the CS doesn't make any biological sense, then it won't be associated. Like if I try to get my dog to associate a flavor with going for a walk, I think that's going to be pretty tough, and we call that association bias. So what does it tell us all? That contingency, or the ability for the CS to predict the US, is critical for classical conditioning, more so than contiguity or even closeness in time. And that's what Rescorla-Wagner model was saying. Okay, that is the end of my mini lecture. I hope you found it helpful.